Okay, hello, good day, everyone, and welcome to today's webinar series. Okay, some people are still joining. Okay, let's just wait for others to join so that we can get started. Okay, by the way, I'm Abdi Yakin Ahmed by name, and I'll be the moderator for this particular program. And I welcome you all to today's session. I promise it's going to be an insightful one. Okay, while we are in the call, can we just make use of the chat box to introduce ourselves? I just type your name and where you are joining the call from in the chat box. Okay, Rizwan Khan, you're welcome. Hello, can you hear me?
Yes.
Okay, thank you. I want you all to just drop your name and where you are joining the call from using the chat box. Okay, Jacinta from Ebonyi State, you're welcome. Thanks for joining. Mustafa Mohammed from Maiduguri, that's Nigeria. You're welcome. Thank you for joining this session. Okay, Swallow Barnabas from, okay, you are joining from Ghana. Okay, thank you for joining. Kayode from the Federal University of Technology, Futmina. Okay, thank you for joining. You're welcome. Chukwu Keneshi from Enugu State, Raphael Ogbe from Makurdi, Nigeria. Okay, thank you for joining. Okay, Shahid Ullah from Pakistan, you're welcome. Welcome once again, and thank you for joining the call. Okay, our webinar topic for this session is natural health and wellness through probiotics, and understanding the genetic foundation of probiotics as nature's gateway to better health and functional innovation. Okay, I'll be introducing you to the host of this particular webinar series, which is Genatures. Genatures is a subsidiary of Genoma Holdings, based in Nigeria. Okay, Genatures, we utilize nature to improve health by harnessing nature's power for sustainable health solutions. And we are a subsidiary of Genoma Holdings, and we utilize African medicinal plants and microbes to profile sustainable solutions for global health challenges by integrating genomics, artificial intelligence, and medicinal chemistry. We are committed to pioneering research in translational medicine and natural healing. Okay, what we do in Genatures, okay, we carry out translational research and development. We also carry out the product creation and commercialization of products. So we have some products that we researched on that we are commercializing and is being in sale right now. Personalized wellness and consultations, innovation and technology integration, education and community outreach, like this particular webinar that fall under the education and community outreach. and strategic partnership and collaboration. Okay, Genetures offers a range of natural products, including the remedies for diabetes, diarrhea, and hemorrhoids, alongside versatile virgin coconut oil, all crafted to promote holistic health and wellness through nature's power. Okay, you can book a session with us and you can reach out to us at www.gnatures.com for more information. Okay, now back to today's business. I'll be introducing our speaker, which is already on the call, in person of Miss Adeshina Christiana. Miss Christiana is a researcher and a mentor that will be taking us on this particular topic. Okay, let's just use the reaction emoji to welcome our speaker on the call, in person of Miss Christiana. Okay, thank you for joining.
All right. Good evening, everyone. Good evening from this side. You are all welcome to today's webinar session. Thank you so much for joining. Thank you so much. All right. I can see some familiar names. To all of you, I say welcome once again. And to those that will be meeting for the first time today, I also say you are all welcome to today's webinar session. And just as we know that we are gathered today because we want to talk about probiotics, probiotics. And so this is a session where we want to share our knowledge. Yes, to share our knowledge and also to gain insights and better understanding as regards this topic. Thank you. I believe we can all hear me. We can all hear me loud and clear. Please, if we can, you can just say yes on the chat box. Yeah, so you can send the reaction. Thank you. And I would like that we all maintain this current silence so that there will be no interference. So after my session, I believe we'll be able to, this session will be about 1 hour, 30 minutes to 2 hours. And then there will be opportunity for everyone to, anyone that wants to also share some insights as regarding this topic. There will be that opportunity at the end of my presentation. So thank you, everyone. Thank you. Okay, so I'll be sharing my slides shortly. I'll be sharing my slides shortly. All right, so I believe my slide is up now. So if you can see my screen, yes, if you can see my screen, you can just send the reaction for me to be sure that I can move on. So can you see my screen now? All right. Thank you so much. Thank you so much. So we will be talking about natural health and wellness through probiotics, and majorly on understanding the genomic foundations of probiotics, as nature's gateway to better health and functional innovation. And when we are talking about probiotics and genomics together, that's we are diving into a field known as probiogenomics, and which is the genomic science behind probiotic function. And our goal for today is to understand how probiotic genes shape health, wellness, and innovation. And whether you're on this call today, whether as a researcher, as a student, a health professional, or you're just curious, I believe that this session will give you a deeper scientific understanding of probiotics. And some people here today that has been with Genetures for quite some time. I believe you know that we majorly deal explore genomics, because just as the moderator made us to understand on what Genetures stand to represent, we are interested in natural wellness and exploring all that this natural wellness has to offer through genomics. And here we are gathered today to explore how genomics has an impact in probiotics. So now let's dive into the main business of today, which probiotics. When we talk about probiotics, they are everywhere today. In supplements, in our foods, when we talk about fermented foods and all of that, and even in wellness products. And even beyond all of this, even we ourselves, we as human beings, we have this probiotics, this bacteria living inside of us. And the reason for good health has to actually is based on these probiotics within us. So, but now the issue is that when we are focusing majorly in terms of maybe supplement foods or wellness products and all of that, the issue is that it's not all probiotic strains that have the same benefits. It's not all probiotic strains that have the same benefits. And the truth is that their real power lies in their genes. Yes, their real power lies in their genes. Their real power lies in their genes. They are not all equal. And how do we know what a particular probiotic strain has to offer in compared to another? It's actually as a result of genomics. I believe you can still hear me, right? Please, I believe we are still together. Are we still together?
Yes.
Yes. The person that said you can't see my screen, I don't know. Probably maybe it might be your network. Yeah, thank you. Please, I would like us to help me do something, because I won't be able to monitor what's happening here. So in case, okay, the moderator will let me know. You can just alert me with a voice if you cannot hear me or if there's a problem. Thank you very much. All right, so I was saying that genomics is that key that helps us to review what a probiotic can actually do. Yes, that's what genomics does. Because when we talk about genomics, it has to do with understanding an organism from the basis of its genetic makeup. And then understanding their genetic blueprint will allow us to make better product choices, stronger research decisions, and as well as being able to make smarter health implications. So that's what genomics, the understanding of genomics does for us. And the truth is that in genomics is actually taking over research in this times. Yes, because the basis of everything is from the gene. It's from the genetics, the genetic makeup. And that's why we are seeing the wealth of information that has to do with making use of genomics to explore probiotics, Okay. So our major highlights for today is we are going to we'll begin with a simple introduction to probiotics and natural health, and we are going to understand, we'll explore the genomic foundations behind probiotic function. We are also going to be understanding some of the essential functional gene categories, because when we're dealing with probiotics, there are some functional gene categories that is peculiar to probiotics. We want to understand some of those essential gene categories, and we are also going to be seeing some case studies. That's where we evaluate real probiotic strains. I'll also be showing us a little live demonstration with a probiogenomics tools, how we actually analyze this probiotic bacteria to gain insights and all of that. So it's actually going to be a wonderful session. Okay, so now what are probiotics? When we talk about probiotics, we are dealing with live microorganisms, which are majorly bacteria, organisms that provide health benefits. And it is found in the gut. That's in our human gut. It's also found in fermented foods, and it's also found in supplements. When we talk about, there is this saying that every disease begins from our gut. Yeah, there is that popular saying that every disease actually stems from the nature of a particular human gut. So what makes us to be healthy, what makes us to be able to digest our food well, has to do with the population of microorganisms. That's the population of probiotics, beneficial probiotics that resides in our human gut. And then when there is an imbalance between the good bacteria and the bad bacteria, that's when some of these diseases comes up. And that's why when we talk about natural health, we cannot but talk about probiotics. Because we ourselves, we are actually made of these probiotics from within. So we need to explore sources. We need to explore natural sources that can help our microbiome community to be strong. And that's the reason. And that's why probiotics matter, because they support immunity, they help us to improve our digestion, and also in terms of production of vitamins. Some of these probiotic bacteria produce vitamins even for our body, and they help us to improve, to stabilize our metabolism as well. So in as much as all of these interesting and wonderful things about probiotics, I want us to understand that the benefits of these probiotics, it depends on the genes inside each strain, not the species name itself. Yeah, it has to do with the genes inside each strain. And when we talk about different strains of bacteria, it can be the same. Maybe, for example, we have Lactobacillus plantarum. And when we talk about Lactobacillus plantarum, there are several strains of this Lactobacillus plantarum, each strain having a unique genetic structure. And it is that genetic structure peculiar to each strain that determines the benefits, its benefit. That's why when you are taking, maybe if we have all of these natural food scientists, if we have them on call here currently, when we talk about maybe you are producing yogurt, you are not just using a bacterial culture just for using sake, let me put it like that, but you are making use of maybe a particular strain. That's in terms of functional food functional food makeup, because maybe there is this particular strain that you see that, oh, it has ability to produce this certain vitamin that another one cannot produce, and then you make use of that particular strain and to produce your yogurt, and then that becomes a functional food to tackle a problem. So that's it. It is the gene within each of the different strains of bacteria that actually determines the benefits that such bacteria, such probiotic bacteria can give. So that means that benefits are not automatic. So maybe because you hear, oh, Lactobacillus plantarum, the benefit is not automatic between all strains. Is not. That's why we need to make use of genomics to explore what are those genes that are present, and then we are able to understand better and make use of them better. Now, when we talk about the genomics foundation of probiotic function, we are talking about how the probiotic genome can help us to determine the survival in the gut. Because even before we talk about the function or the benefits that that a certain probiotic has to offer to man, not even man, even talking about animals, because even the use of probiotics is now being used in feed formulation for animals. So before we even talk about that function to whoever is going to benefit from it, we have to talk about the probiotic itself, how well it can survive. How well it can survive. So they also need genes that can help them to survive in the human gut. Because in the human gut, we can talk about maybe how well they can survive to heat, or how well they can survive to the bile. That's the bile in the human digestive system, or how well they can even survive to the stomach acid. So even the probiotic bacteria itself must have genes strong enough to help it survive in the gut. So that is the genomic foundations. When we begin to explore this probiotic bacteria from the genomic level, we can see how well such probiotic can survive in the human gut. Talking about adhesion to human cells, how well they can fuse with the human cell. Yes. And also then we now talk about their ability to produce vitamins, then their antimicrobial defense, how well they can defend against other harmful microorganisms, or even talking about in terms of making use of antibiotics. Because when we make use of antibiotics is it actually functions to destroy whether the good bacteria or the bad bacteria in the human gut. And that's why it's advised that when someone the abuse of antibiotics, and when someone makes use of antibiotics, you need to take foods that help you to grow back your good bacteria. And so we need probiotics that can even withstand some of these antibiotics as well. And then through genomic foundations, we can understand immunomodulation. That's how a particular probiotic can affect the immune system. Yes, so that means genomics can help us to even understand what this probiotic can do even before we make use of it. So I've talked about stress tolerance and all of that. So in summary, we say genomics is that scientific way to know which strain offers which benefits. It is genomics that makes us to understand that. The scientific way to know which strain, which bacterial strain, which probiotic strain offers a certain kind of benefit. And this is actually the basis of when we talk about personalized nutrition and all of that. This is the basis of it. This is the basis of it. Okay. So now, why genomics is the future of natural health? Why genomics is the future of natural health? Why does genomics matter? Why does it matter? You know, genomics helps us to move beyond the marketing claims. Maybe, okay, you get a certain product, it says, okay, this product is good for this, is good for this. We can test it beyond the claims. Or even you as a maybe a food scientist, you can back up your claims with the help of genomics. You can back up your claims if you say that this particular food works for this. You can back it up with true scientific measures, and that's that's why genomics actually matters. Yes, genomics helps us to reveal the functional traits and not just guessing and not just guesswork. That's what genomics helps us to do. And then it also helps researchers. It's very essential for researchers and developers to build better probiotic products, and it even helps to educate the consumers as well. It helps us to choose strains with real benefits, not just about maybe, you know, when we talk about bacteria probiotic and all of that, there's this CFU. I think that's the colony forming unit. Is beyond all of that. The benefits that a probiotic has to give is beyond just the colony forming unit and all of that. It has to do with revealing the functional traits that is dependent on different strains of bacteria. And then, just like I noted before, genomic matters because it supports personalized health and nutrition. It supports personalized health and nutrition. Okay, and please, I believe I'm not too fast. I hope I'm not too fast. Are we following me very well? Are we
Exactly, ma.
following me
Yes, ma.
very well?
Yes, ma.
Thank you. Thank you so much. Okay, so when we talk about there are some key functional gene categories in probiotics. Some of those genes, when we talk about genes that are responsible for gut stability and adhesion genes, this has to do with how well the probiotic itself can survive, the gut stability. So we talk about mucin binding proteins, we talk about adhesins, we talk about biofilm regulation genes. All of these genes helps the probiotic itself to stick to the intestinal lining and form stable populations. So those are some of those genes responsible for that. How well the probiotic can become stable in the human gut. And then we talk about genes that are responsible for immune modulation. Genes that are responsible for immune modulation. These genes affect how the probiotic interacts with the immune cells, either calming inflammation or boosting defense. So there are genes that are responsible for this, immune modulation genes. We also have the vitamin production genes in terms of genes that are responsible for folate production or vitamin B12 and all of that. So some probiotic strains can naturally produce these B vitamins, including folate. And that's the reason why we have to make use of genomics to assess these strains. How well can strain A produce vitamins compared to other strains? Or maybe I think we just concluded this phylogenetics. There was a workshop we just concluded talking about phylogenetics, and in that class we were talking about how, okay, you sequence your organism. You sequence, maybe for example, you sequence your bacteria So in this case, I'll be likening it to maybe you sequence you discovered a new strain of probiotic bacteria, and then you want to compare your strain with other strains in terms of, okay, how better is this strain compared to other strains that are available out there, and all of that. So those are some of those comparisons that we do through genomics, and then we talk about the antimicrobial safety genes. There are genes responsible for this antimicrobial and safety genes in terms of the bacteria itself, the probiotic bacteria itself, how safe it is for us to incorporate it into food. How safe. We need to check if there are hoping that there is absence of harmful virulence genes. So through genomics, we're able to see that a probiotic bacteria that will be used for food will not contain all of these virulent genes to ensure safety. So those are those things that we see. And why these genes matter? Why do they matter? These genes, they determine whether a probiotic bacteria can survive the stomach acid, remain active in the gut. When we talk about now, when you analyze a probiotic and you see that a particular probiotic has weak adhesion genes, it can't stay long in the gut and it will have no effect. Yes, something that cannot establish, should I say establish its authority? Yes, let me use that word. Something that cannot establish its authority in the gut will have no effect. So that's why all of these genes matter. A strain that does not have vitamin production genes, it cannot contribute anything to the micronutrient synthesis. It will have no contribution in terms of nutrients to the host. Yes. And if a strain is also lacking in antimicrobial genes, it will not be able to compete with harmful microbes in the gut. It will not be able to compete. And then, so that means that with all of this, we have been able to see that the absence or presence of a gene will determine the kind and extent of the health benefits of whatever that probiotic strain claims to offer. Yes, the absence or presence will determine the kind and extent of health benefits that a certain probiotic has to offer. Yes. So how does functional genes translate into real health benefits? So okay, when I see that this gene is present, how does it translate into real health benefits? When we talk about bacteriocins are, there are some kind of how do I put the word now? Okay, bacteriocins, they are things that are synthesized by some of these probiotic bacteria, and they are synthesized to actually fight harmful microbes. So a particular probiotic bacteria, when it's in a community of harmful microbes, it can synthesize these bacteriocins to fight these harmful microbes. Or even beyond that, even if you are talking about even food preservation, how can you preserve a food? Maybe those people that are into all of this microbiology and all of that, talking about food preservation, even the insight, the understanding of this can help you to formulate preservatives, natural preservatives. Because okay, when you see that this particular probiotic strain produce, now when we talk, there are different kinds of bacteriocins. We have them different kinds. So when we carry out some of these analysis and we're able to see that, okay, this is the particular bacteriocin, this is the particular bacteriocin that this probiotic strain is producing. It will help us to know, okay, it can fight, help us to preserve this food better. I think, okay, let's do a little check. Let's do a little check on probiotics, sorry, on bacteriocins, different kind of bacteriocins that we have. Different kinds of bacteriocins that we have. Okay. All right. So can we see this? Can we see some of this? Now, when we say bacteriocins are bacterial produced antimicrobials, classified mainly by their structure. We have like class 1, class 2. Class 1 bacteriocins are antibiotics, and we have mycin as an example of a bacteriocin under the class 1 bacteriocin. We also have class 2. Class 2, we have some of them like pidoicin, elveticin, and all of that. We also have, now the function of all of these bacteriocin is that they kill or inhibit other bacteria by damaging cell membranes, interfering with DNA protein synthesis, or disrupting cell walls, making them valuable for food preservation, like the niacin, pidoicin. And they are also potential therapeutic agents against pathogens. So these are some of those beautiful things that we see. So I can analyze a particular probiotic and I see that, okay, that probiotic is producing pidoicin, or it is producing niacin, or it even has the gene that produces both. So I will now know that, okay, because of this, I can make use of this particular strain in terms of for food preservation and all of that. So that's some of those beautiful things that probiotics has to offer. Please, are we learning something? Are we learning something? Yeah, I believe
Yes,
we are learning something, right?
Yes, ma'am.
Yeah, thank you. Thank you so much. Thank you so much for that. Thank you. All right, so I go back to our slide. So we've talked about bacteriocin. We've then talking about adhesion genes. Adhesion genes. Adhesion genes will help the strain to colonize the gut. Because something, even if it has all of these wonderful things to offer, if it cannot colonize the gut, then it's actually of no use. All of those interesting things it has to offer cannot be actualized if it does not have enough genes that are responsible for adhesion. So every probiotic bacteria, we must have these adhesion genes. Adhesion genes very well. Yes, let me put it like that. And then you talk about vitamin genes. Vitamin genes will help us to will contribute to supporting nutritional health. And then we talk about stress response, how well the bacteria itself can respond to stress in terms of surviving through harsh conditions of the human gut, like acid, bile, and all of that. It has to have enough genes that will help it to survive all of those harsh conditions. And then, of course, immune genes that helps to fight against inflammation and all of that. So with all of this, can you now see how genomics is actually that bridge between probiotics and real wellness? Real wellness. That's the bridge. That's the bridge. It has a lot to uncover. It has a lot to offer. Yes. So let's talk about how to analyze a probiotic genome. You know, you can get for example, you can practicalize getting maybe a genome from available strains that are already available on the database, like the NCBI, or even maybe you carried out a sequencing yourself. You carry out the sequencing yourself. You run some annotation. There are different bioinformatics tools that we make use of to run annotation on probiotic genomes. and then you now identify functional categories. When we talk about functional categories, those are all of these things that we talk about, like vitamin category, stress response, adhesion, and all of that. So you can now do what? You can also compare with other strains. You compare with other strains, you extract, you interpret the genetic profile. So when you carry out all of this, you identify all of those functional categories that are available. You now do maybe like interpret it. You carry out the genetic profiling, and then you can now apply whatever insights you get in probiotic, in product development or even research. So I noted that I will show us a little bit of some of these things I talked about here. So let me hold on this. And when we get into, I'll show us how one of these annotation tools that we use in probiotic research. And then another important thing I want to emphasize is on the safety assessment, because we need to assess safety through genomics. So it is very, very crucial. That's why I want to emphasize it again in terms of probiotic science. So genomics will help us to check genes that are virulent. This gene, hope there is no virulent gene hiding within it, or mobile gene elements that can carry harmful traits. It's not as if it's the bacteria itself that has that trait. But hope there are, maybe when we talk about all of these plasmids and all of that, we want to be sure that there are no mobile genetic elements. Because when you talk about mobile genetic elements, how that a particular gene can be transferred from one bacteria to another. So we don't want things like that in a safe probiotic. And then we want to understand that a safe probiotic will we want to check antibiotic resistant genes. When we talk about this antibiotic resistance, for the bacteria itself, in terms of its surviving the gut, it actually needs these antibiotic resistance genes. Yes, it needs it for its own self to survive. And that's why we want to make sure that there are no mobile genetic elements that because there are some things that are like our housekeeping genes necessary for the bacteria itself to survive. So then we want to be sure that these mobile genetic elements I was talking about, that this antibiotic resistance cannot be transferred to other neighboring bacteria around. So those are some of the things that we check, safety assessment, and then we want to see that the probiotic bacteria it lacks toxic genes. So that's why a responsible probiotic development always include genomic screening, because you cannot make use of a probiotic bacteria and then you use it in food formulation and there is no genomic screening. It's not possible, because it's from genomic screening that we are able to be sure indeed that it is safe to use. And that's the beauty of genomics. That's the beauty of genomics in exploring natural wellness through from probiotics. Okay, so we have some of these case studies. I think I have some journals that I will share with us. We can read some of these things. We can be aware of some of these things that we are talking about here today. There are some research that has been done on them. So I'm going to, in whatever group that we are, from whatever WhatsApp group you join, I'm going to be sharing with us some of these research papers, some of these journals with us. Okay, so there is a lacticacea bacillus ramonis. So even this it's a species Even within this species, we have different strains. We have different strains. So, but in generally for this particular species, Lacticacebacillus ramonus, it has been known to have strong adhesion genes like good pili and all of that. It has been studied to have these good adhesion genes for gut stability. It has also been studied to produce bacteriocins in terms of antimicrobial activity and all of that. So it is capable of all of this. It is capable of all of this, and it is a very strong gut health probiotic. That's on a general basis. On a general basis, if we are now doing strain by strain in terms of this Ramonus, a particular strain might be stronger in one of these than the other in terms of the different strains of this particular species. Same with Bifidobacterium longo. Same with it. It also have different strains, but on a general note, it has been known to actually have it has very wonderful vitamin pathways, production of folates and all of that. It has very good stress tolerance, and then also anti-inflammatory genes. It's also this bifidobacterium has also been known to even for when we are talking about infant health, is very beneficial for them. So the place of now comparing a strain to another strain is how well is this particular strain A stronger than strain B and all of that. So that's it. That's it. Now, where probiogenomics is used today, the word probiogenomics is actually the application of genomics in probiotics. So that's probiogenomics. Is a skill on its own. It's a skill on its own that is actually very relevant in this current time. So it's not just academic. It's very practical. It's applied in designing functional food, in designing, in developing nutraceuticals, also in terms of the knowledge of it can be used in personalized nutrition. We talk about medical probiotics, microbiome-based therapies, research and clinical studies, and all of that. So this science of probiogenomics drives innovation in wellness and microbiome industries. I don't know. I really want to know. Let me get to know some of us. Anybody from that is actually exploring this field. Maybe you are a student in the field, or you are already exploring it. I want to know us. I want to know us. Can we just tell anybody in this field? It would be interesting that to also hear from us. So I want to know some of us. Anybody in this field? Anyone? We can just signify on the chat box. Anyone? Anyone that deals with, okay. So Teresa, after my class, I'll be interested to hear some things from you as well. Anyone else? Anyone else? Or perhaps maybe you are a student. Maybe you are a student coming up in this field. You can also just signify as well. Okay. So I would like to hear from us as well. I would like to hear from us as well. Oh, beautiful, beautiful. A nutritional biochemist trying to switch to nutrigenomics. This is very beautiful. Okay, your questions. We're going to address question after I'm done. Okay, a student of aquaculture having interest in fish genomics. Beautiful. Of course, even this, what you are learning, you know, I mentioned the fact that even probiotics is now being incorporated into animal feed and all of that. So you can come up with something beautiful in that aspect of fish. It's to be a very wonderful one, a very wonderful one. Okay. All right. So let me move on. So that's it. That's it. Okay. So when we now talk about the future, we're already in the present, and there's also a future of this probiotics, just like some of those beautiful things that you all have introduced. Someone that said wants to switch into nutrigenomics and all of that. They are beautiful, very beautiful, and this field is actually advancing very quickly, very quickly, very quickly. You also want to position yourself. You also want to position yourself in this field. You also want to position yourself accurately in this field, gaining these practical skills of probiotics. Because when we talk about, there are now AI. AI now helps us to even predict probiotic function from genomics, from genomes and all of that. Customized probiotic formulations, like someone that is talking about personalized nutrition, nutrigenomics, and all of that. There's also precision microbiome health. Precision. You are very precise about the health of, because what works for what a particular patient needs might not be the same thing that another patient needs. So all of this helps us to be precise in our in our health and all of that, in our diagnosis and all of that. So there's also functional gene engineering, next generation probiotics. So there is a whole lot of opportunities in this field, a whole lot, a whole lot. And that's the reason why a webinar like this is coming up, that can equip us even to position ourselves in this future. It's a very huge field with huge potential for future health solutions. Yes, it has a whole lot to offer, and we also want to position ourselves. And my advice to students here on this call today, one of the beautiful things that that really helped me, because I think I became exposed to genomics during my 400 level days. Yes, I became exposed to genomics when I was still an undergraduate in 400 level, because I did my industrial training in this genomics. So that was when I got to, and because of that, just that one experience, it has really helped me in my field, in my career, and all of that. So it's an advice to students, beyond just the certificate. Okay, yes, I graduated as a nutritional scientist and all of that. Beyond that, it's an advice that you also pursue skills that can make your certificate stand out better than others. What can make your own certificate better than others is when you pursue skills that are relevant to whatever field that you are studying. Yes, I believe we all know that it's beyond just the certificate. It's beyond just the certificate. It's the skills that can really put us on that platform. So that's a little advice to, maybe if you're an undergraduate here, I will advise you to pursue skills like this, and all of that. Okay, thank you very much. So in summary, the key takeaways that we are learning from here is that probiotic benefits, it depends on the gene. It's not just the species name. It depends on the genes that they have. It's not just the species name. And genomics helps us to reveal this true probiotic potential. And then we understanding the key functional gene category will improve health decisions. And then there are simple bioinformatics tools that make probiogenomics very easy to explore. Very easy tools. I will show us that shortly after now. And then this knowledge, in summary, will help us to empower better choices in research, in product development, in personal wellness, and all of that. So that's it. Okay. So let me show us some of these things I was talking about. Um, okay. Some of the analysis I've done before. So this is a bioinformatics tool called Rust, used for annotation. Used for annotation. Okay. I think there is a little issue with the site currently. Just give me a few minutes. Let me make use of another link for that. Um, okay, I think there's a little bit of issue currently. I can't seem to access it from my laptop currently. I would have really loved to show us this. I'll have really loved to show us this. Okay, let me do it this way. Pending when I'm able to log in from my laptop. Oh, okay. Can we still hear me? I think I just had a little switch. So can you hear me now?
Yes, ma'am.
Yes.
Yes,
Okay,
ma'am.
okay, thank you. Though, let me see. The screen here might not really be very visible, but let me just try it. Let's just try it here. Okay, pending when I'm able to rectify that on my laptop. I don't know. Can we see what I'm showing currently? RAST, that's the rapid annotation using subsystem technology. Can we see it?
Yes.
Okay, so this is it. And these are some of those. Wow, I think it's looking like it's the same issue here. And just a few hours ago, it was showing. So I think it has to do with the site currently. It's not about the location. Wow. Okay. So I think the site currently has a problem. I would have really loved to show us this. Wow, 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 wow. Okay. So since we can't do that, we can't do that now. We can't do that now. The site really has a problem currently. I need to switch back. Sorry, just okay. So I'm back. Um, I don't know what's wrong with the site currently. All right. So apologies everyone. So since the site is currently probably undergoing some kind of probably maybe they are undergoing some kind of maintenance. So I will not be able to show us some of those things I wanted to show us practically. Yeah, I won't be able to show us that. So, but nevertheless, okay. So I think I will have to stop here. I will have to stop here. That was just the last thing I wanted to show us. How different things as regarding the when I did some analysis between different strains of the bacteria and all of that, and some of those results, I wanted to show us how the result looked like. Okay, so since we can't do that currently, I will want to pause here. Probably maybe maybe before we finish, probably maybe I might have even been able to rectify that. But if that's the case, let me go over to those people that I want to also share some insights. I think what was that name again? Okay, my name on the group. I think that should be additional Christiana on the group. What group are you joining from? Don't worry, you can just message any of the admin to get to me. But my name is additional Christiana, so you can just go back to your group and check any of the messages that we've sent as regarding the program and all of that. You should be able to locate my name from there. So I want to, because I had to switch between device, all of the charts has disappeared. So there was a particular name I wanted to also share some insights as regarding her field and all of that. Maybe some of those things that you want to comment on what I've shared, what I've thought, and all of that. Or just anyone. Is anybody interested to do that? So you can raise up your hand. Okay, I think, was that Teresa? Someone said some of them are not on the group, on the WhatsApp group. Mr. Med, please, are you still on the call so that you can help us to drop any of our community group so that those that are not on the group currently can join? our WhatsApp community. Our WhatsApp community. Yes, we are going to do that before the class ends. Before the class ends. So anyone, okay, I can see Samina Ejaz raising up, is it his or her hand? Sorry. So you can go ahead. You can go ahead. Okay, Samina wants to talk. Hello.
Yes, ma, we are hearing you, ma.
Oh, okay. When you were raising up your hand, I thought you wanted to also share something. Okay. So I want to hear from people that are probably practicing in this field.
Hi,
Do we have anyone like that?
thank you very much, ma'am, for very nice presentation. I just want to mention that
Is someone talking? Is that Samina? Okay. So Samina, I think you are the one talking, right?
yes, I'm talking, but unfortunately, can you hear me? Can you
Yeah,
hear
I
me?
can, but your voice is quite a little bit down. So if you can help us to increase your voice a bit.
First of all, ma'am, it's very nice presentation and very. I got many points from here, and I want to mention that would it be possible for you that we can have the presentation, a copy of presentation or the recording of the meeting of today? And second is that you mentioned that these probiotics, they have the genes that confer them to bear stress resistance. So I just wanted to inquire that what type of stress they can mm, what type
Okay, you
of
said
stress they can
talking
bear.
about what type of what?
Stress related genes. You talked about the stress related genes.
Oh, okay, stress-related
So I want to
genes.
know about something that what kind of stress they can withhold.
Okay, I talked about the fact that some of those stress, that is in terms of the stress in relating to the bacteria itself
Yes,
surviving. That's
yes,
the
yes,
probiotic bacteria.
yes.
Surviving maybe the stomach acid, bile, and all of that. So it needs genes that can help it to survive all of those. Those things are stress. Heat is
Okay,
stress. I believe we know that probiotic bacteria, um, when they are exposed to some certain level of heat, I think they lose their benefits and all of that. So that's what I meant by stress. So their ability
okay,
to be able to survive in the gut. So be it acid stress, it's really painful currently that I what I wanted to show us. I can't show it currently due to the site being down, due to the site being down currently. I will have really showed us
okay,
functional categories. And then you talked about the recording. Yes. This session is live streamed on our YouTube channel. So when you have the YouTube link, you can always go back at any point in time to watch it. To watch it.
okay. Thank you very much. Thank you very much.
Yeah, thank you so much. Thank you. Okay, someone said, can probiotics modulate metabolic disease? Yes, yes. I think I should be able to send some of these journals that I've come across as relating in terms of research, um, in terms of this obesity and all of that. Of course, we have probiotics that produce some of these things. Even beyond diabetes, obesity, even in terms of when you talk about neuro, that's the brain, gut-brain axis. There are also probiotics that even have benefits when it comes to do with the nervous system and all of that. Do they need refrigeration? Do they need refrigeration? That's bacteria itself. We have different. That's why we talk about the different strains. There are some that need such kind of environment. So that answer to the question will determine whatever thing that you get from your research, and then we determine, okay, if this needs refrigeration, if it does not need, and all of that. If it does not need, and all of that. But generally, probiotics, they don't like extreme heat. Extreme heat can kill their function and all of that. So it's a kind of moderate temperature. Moderate temperature. Okay, isolating, you're interested in isolating specific genes from the gut. Could you please advise on the best approach? Yes, in this aspect, because when you talk about isolating specific genes, when we are dealing with this is a kind of metagenomic study, a metagenomic study, because when you say specific gene, you might not know what, you can't really say the specific thing you want to isolate from the gut. Let me put it like that, because they are different community. So when you do a general microbiome metagenomic study, in terms of when you get the gut sample, every available organism in that gut sample, you are going to get it from, okay, you sequence the gut, so they are diverse. So you will now check from the diverse results. That's when you do other sequencing, and then you now do molecular characterization and all of that. So I want to refer back to the phylogenetics class we just recently concluded, when I was telling the participants about what's the word? What's that again? Molecular characterization, molecular identification. That's actually the pathway of bioinformatics research. You carried out your sequencing, you want to identify. That point of identification is molecular characterization. And I also wanted to say that some of these programs that we have, some of them that were free, we still have them on our YouTube channel. So you can go back to our YouTube channel and you can watch some of these things that I'm talking about, because I think we practicalized it in. So there was a free session where we did this molecular characterization as well. So the link to our YouTube channel will be dropped. You can go through it. You can go through it and learn some other things. Yes, you can learn some other things. So when you carry out the molecular characterization, you get it's like you carrying out blasts. Yes, basic local alignment set 2, and then you now get the result of all the available bacteria that you got from the gut that you sequence. So it is at that point you will know which organism is in the gut, and then you can now find it, okay, whether if your specific gene of interest was among them. And that's, let me use this to also talk about some of these deficiency. Maybe there's a particular patient that is suffering from something, and then you now want to understand how the gut microbiome, the community. From that thing that I just explained, you'll be able to know a particular bacteria that is present or absent. It can be as a result of the absence of a particular bacteria in the gut that is causing that disease. So because of that, you now see that, okay, this is absent, then this person needs this. And then you now formulate food for such kind of person to introduce that absent bacteria. Or maybe the bacteria is not available in sufficient quantity. So that's it. That's for like someone that was asking me about metabolic disease. It's something like that as well. Something like that. Okay. So our WhatsApp community has been dropped. Please, can we do well to join now? Those people that said they are not on the chat, because every other information, every other things that we want to send, the YouTube channel, the some of the research papers will be sent in the group. So it has been dropped. Please, you can do well to join. You can do well to join now. So let me see. Okay. So Ajani Kayode would like to talk on fish genomics. Very interesting. I also want to learn some one or two from you. Thank you. Before that, let me answer this question. What is the inflammatory role of probiotics in case of chronic symptoms of certain diseases such as elephantiasis? The anti-inflammatory role, okay, in case of chronic symptoms of certain diseases such as elephantiasis. This question has to do with it has to do with, first of all, I want to understand what is this elephantiasis. What are some of those what causes it? Yes, elephantiasis. That's swollen leg, if I'm correct. Yes. So I want to understand because for me to understand the anti-inflammatory role of probiotics in this, it might not even be elephantiasis. It might be another thing. I also need to understand the genomic basis of this disease, because understanding genomic basis of a disease and then understanding genomic action of the probiotic, they go hand in hand. They go hand in hand. Even if I might not be able to answer this question exactly the way you might want it currently, but if I want to understand this, of course, I want to go and understand about elephantiasis itself, and then I now enter into the genomic aspect, understanding what are those underlying genetic causes of it, genetic actions of it, and then I now carry out my research on probiotics, some of these probiotics that might have this anti-inflammatory role, because gene affects gene. Let me put it like that. Gene affects gene. So I want to do my study on the genetics of the probiotics, whatever probiotics I might want to be making use of, and all of that. And then so that's it. That's just it. I don't know if I answered your question a little bit. So let me hear from Ajayi Kayode on fish genomics. You can go ahead.
All right, thank you, ma. Thank you to the G Nature. Okay, I'm still an undergraduate student in final year, and the trending now is on fish genetics and biotechnology, especially on genomics. The use of genetic information to improve the growth and disease resistance of fish population. You know, the main issue that we are facing in aquaculture now is there is much demand. There is much demand, and to meet that demand with limited resources available now is quite a thing. So, researchers in aquaculture, they are now tending towards checking the genetics information of fish and how they can get to improve on if they feed on small quantities of feed, how it will improve their growth,
Okay.
and also to check on the disease resistance from bacteria, parasites, and the rest. So how to conquer them. While they are doing that, they are still trying to check the ethical implications of everything. So it has been quite a thing. But as undergraduate students in Nigeria university, public university to be precise, it's quite tough with low infrastructure and financial constraints. So I'm quite incapacitated to, but I'm looking towards the opportunity to just learn more on molecular biology, just to understand some things and um, get to do some analysis on the platform like Blast and the Rust that you you did now. And also I know about fast skill, but I've not really been getting to do much on them. So I don't know if there's anything that can be of help from you for me, sir, for me, ma.
Oh wow, that's a very beautiful one. I love what you said. I love what you said because um in terms of, okay, you talk about Nigeria and all of that. So even with that, it doesn't still limit us actually. So just like you noted, and you equipping yourself with the skills and all of that, and then as opportunity, because you don't get prepared at the point of opportunity. You already have the you're already equipped, and then when opportunity comes in like this, you just make use of it, maximize it. And even with before infrastructure and all of that, there are still things we can do, purely bioinformatics aspect. And because of what you said, actually we hope that beyond just this theoretical aspect, that you can also dive deeper into these skills learning these skills, like gaining the practical bioinformatics, how to really analyze, introduction to the tools that is being used, how to you see it practically. So even after this webinar, we still want to introduce a workshop. Actually, that one will not be free. That's just it. That one will not be free. You have to just should I say, you are going to pay a little token of commitment fee, even to gain hands-on experience on all of this. And that's one of the reasons why we really want to push out this all of these all of these skills all of these skills. And that's the reason why that's one of our burden in terms of educational outreach, really equipping researchers out there, students out there, to gain all of these practical skills, to gain it. Because learning things like this, the just like you noted when we were talking about infrastructure, even financial constraint regarding all of this. These skills that we are pushing out, in terms of learning them, it's a whole lot. It's a whole lot. But we always want to bring it at a very subsidized rate, because beyond just the money, what is really driving us is impact. And so that's why we bring skills like this, we bring trainings like this that are at a very affordable rate for everyone to be able to participate. So if you still want to learn more on this, there will be we are going to be advertising a workshop. Yes, we are going to be advertising a 3 days workshop where we explore this in total. Yes, we really explore this. And the beauty of it is that it's very beginner friendly. All of this, just you said you have insights to Rust before and all of that, but you don't really have to use it very well. All of those things, we are going to be exploring it. There is, even beyond Rust, there is another bioinformatics tool that is specifically used in checking for this bacteriocin. So it's a separate tool on its own for us to know what's the bacteriocin that is in. So they were going to explore a whole lot of those tools, and all of that. So if you want to gain more on it, so for now, I will just say, immediately after this webinar, we are going to drop another link on this, or perhaps we can just drop it here. if you want to learn more as regarding the practical aspect of analyzing all of this, I think it was at this point where I was talking about it. Yes, how to analyze a probiotic genome. So that workshop is actually we are going to do all of this thing practically. So if you are interested in it, you can just join the group. You can just join the group. So I think Mr. Ahmed, you can also help us to drop the workshop group as well for those that are interested to learn more about it. The curriculum for the workshop will also be dropped, and you can gain this skill even before the year runs out. Yes, even before the year runs out. Okay. So from Timothy, my contribution and future prospects. Okay. So I'm looking at the possibility of isolating these genes and introducing them into E. coli. The three basic genera for these probiotics are Lactobacillus, yes, you are right, Bifidobacterium, and Saccharomyces, very correct. One factor that determines effective of this probiotic is dose. Yes, that's true. Small amounts might not work. Since E. coli has high proliferation rates, we can produce thousands copies of these genes, thereby improving health and functional food. This is a very beautiful insight, a very, very beautiful one. Let me see. I think okay. Let me see. Functional gene engineering. This is sounding like functional gene engineering. That's in terms of in producing 1,000 copies of those genes, 1,000 copies of those genes, which is also one of those things that we see in that raster I wanted to show us in terms of because for each of those categories, we call them subsystem categories. A particular, maybe a gene that is responsible in producing this bacteriocin, maybe this strain has maybe 20 of it, 20 of that gene, and the other strain has maybe just 10. Of course, in terms of priority, you might want to prioritize the one that has more genes. So those were those things I really wish I could have shown us. So that's it. What you said, talk about that's under the aspect of functional gene engineering, and it's a very beautiful one. I'm very interested to see how you go into this. It's very interesting. Okay. That with weak immune system should not use this because of opportunistic pathogenicity. Yes, of course, of course. A harmless bacteria, of course, you are very right. You are very right. Which is also linked with one of those things I was talking about on mobile genetic elements. So that's why genomics, it's a whole lot of things for us to really scrutinize even before we make use of them. So that's why genomics, we don't just go on guesswork. We really need to ascertain. So thank you very much for that, Timothy. Thank you so much for that. I really enjoyed everything you said. Okay, so is Samina, do you still want to say something? Do you still want to say something?
No.
Oh, Isham has been raising up his hand. Isham, can you go ahead?
Okay, good evening.
Yes, good evening.
From Nigeria. I actually asked a question earlier in the chat box regarding the anti-inflammatory role of probiotics in the case of disease such as elephantiasis, which causes swelling links, as in the case of chronic symptoms.
Oh, okay. Sorry, I can't seem to hear you very well. Sorry, please just give me 2 seconds. Ishan, you can go ahead. I have a little bit of distraction. Sorry, you can go ahead.
I said I asked a question earlier regarding anti-inflammatory role of these probiotics, as in the case of certain infectious disease such as elephantiasis, which causes swelling limbs.
Okay.
It's it is caused by a parasite called Wuchereria bancrofti. Even though there are different species of parasites, I think there are three that causes this elephantiasis.
Okay.
They actually affect the lymphatic system, and the infection is caused, I think, in 3 stages. The lymphatic system, the adult forms, when they die within the lymphatic system and they keep accumulating, that's what triggers the inflammation. And the other part is they compromise immunity, paving way for other microorganisms or other bacterial infections, which also leads to this inflammation. So it is a combination of bacterial or other microorganisms causing infection, which might lead to this inflammation, as well as the adult worms accumulating with the system after they die, which causes this, which triggers this inflammation.
Wow. Okay. Okay. So I want to ask you, this elephantiasis, is there a cure for it? Like currently, is there a cure for elephantiasis?
No, there for it. It's only chemotherapy and vaccine.
And vaccine. Okay, I understand you. So, in terms of, so that means there's still a lot of research and that needs to be done on that aspect. But the little I just wanted to add was that just like we have different health challenges, health problems, and all of that, in terms of maybe we talking about probiotics now. We're talking about probiotics, maybe in terms of functional food and all of that. And even all of those chemotherapy you talked about, even the immune system of such kind of person also will play a role. So if we are talking about probiotics, maybe we can even begin at least from a point where the kind of food the person eats that helps to still improve the immune system a bit, even in all of those things that person as is undergoing treatment and all of that. So we might want to start talking about the point where eating foods that can boost immunity, and that's where probiotic can now start coming in. So that's just a little addition I wanted to add on that. Yeah, I don't know, do you get me?
Yeah, I got it.
Yeah. Okay, thank you. Do we
Actually,
have anyone else that wants to say something? Maybe a contribution, a question, anything. The floor is open for anyone. I think as we probably maybe one or two persons more, and then we'll be rounding up the session. Please, I believe everybody has joined the WhatsApp group. It's very important because as soon as the call is over, you won't be able to join the link again. So please, if you have not joined the link, you can join. You can join the link. You can join through the link. Okay, so do we have anybody else? Okay. I just saw a question from from YouTube. I love this. How stable are probiotics during storage? And do they still contain the claimed CFUs by the time I consume them? This is very interesting because uh, um, understanding understanding the genetic makeup of different bacteria is also one of those things that we find out. Being able to know how stable the shelf life, all of those information is still from all of this probiogenomics analysis. We are able to know. We're able to know in terms of stability, yes, because I was talking about some of those genes that helps the bacteria itself in terms of adhesion. So there are different functional gene categories. There are different gene categories that that helps a bacteria. So it is based on the genes that are responsible for that stability that will now determine maybe the shelf life. The shelf life now in terms of okay, maybe does it still have the claimed CFU? So if there's a bacteria that has, okay, this, like I was saying the other time, maybe this one has 20 genes responsible for it, the other one has 10, you will know that, okay, the one that has 20 genes in terms of priority, I want to go for that one that has 20 genes because the shelf life will be higher. And it's from that we are able to those things. When I'm saying 20 genes, this one has 10. That's what we see practically during the analysis. So that's it. That's it. That's it. So okay. The major roles that are played by probiotic in the body. That's another question from YouTube. Major roles that are played by probiotics in the body. Now, when we talk about the major roles, okay, let me just give you some number one function, of course, is nutrition. Because when we deal with nutrition, in terms of enhancing digestion, you know, it is some of these bacteria that helps us to break down our food more efficiently. So in terms of enhancing digestion, and when you're talking about even probiotics itself, there is another terminology in this field of probiotics, science that has to do with prebiotics. There is pro and there is pre. When we talk about pro is the bacteria itself, the bacteria itself, and then prebiotics are some of those foods that you can eat, like that can help to feed even the probiotic itself, because the probiotic also needs to feed to grow. And so there are some foods that you can recommend together with whatever probiotic food that you are giving. That's the prebiotics that helps to also increase the growth of the probiotic bacteria in the stomach. And then so under nutrition, on the functions of the probiotic, I've talked about enhancing digestion. It also produces SCFAs. SCFAs, that's talking about short chain fatty acids and all of that. And then probiotic also help us in absorbing nutrients. You know, it's one thing to break down the food. It's another thing to absorb. It's another thing to assimilate. So some of these probiotic bacteria also helps to improve nutrient absorption. That's maybe absorption of minerals like calcium and all of that. So probiotic also play those roles in nutrient absorption. Then the production of vitamins is also there. Production of vitamins is also there. So that's all under nutrition. Another role that probiotics can play is on immune modulation. immune modulation. There's something called leaky gut. When a gut is leaky, that's the gut wall, the intestinal walls is not very strong, and there is a disease that's associated with all of that. This IBS, inflammatory bowel syndrome, all of those diseases has to do with maybe when the gut is not strong enough. So in terms of probiotic helping to modulate the immune system, it can help to strengthen the gut bacteria, to strengthen the gut barrier. The gut barrier. It also helps us to stimulate maybe protective immune response. Maybe it can activate immune responses. It can activate immune responses or even deactivate. Maybe when you're talking of that diabetes, obesity. So there are it's as a result of some of those immune system that are being activated. So when we take these probiotics and all of that, they can also help to modulate the immune system. All of those systems that were triggered, that is now causing the inflammation, we can get some of those benefits. And is at the point of where we analyze the genome that we will know we are going to know which type of immune modulation is this particular bacteria likely to affect. So it's at the point of that analysis that we get all of that. So it also helps us to fight pathogens, of course, reduce allergies and autoimmunity. I talked about the gut-brain axis the other time, neuromodulation. So it can interact with nerves, different neurons linking to the brain. It can even help in producing neurotransmitters. I don't know if we know that even our mood per time, maybe someone is depressed, it also stems from the gut. Yes, our mood is sometimes what's that word again? Okay, is controlled from the gut. Yes. So we can even carry out this research on some of these strains that can help in producing these neurotransmitters that helps us to influence mood and behavior. So probiotics has a whole lot to offer. A whole lot. I talked about even food preservation itself. Okay, that one now it's not in terms of the host as well. So those are those things that probiotic, it has a whole lot to offer, and that was just little out of the many things that it has to offer. So I think in the absence of any more question or interaction, I believe that this would be a good point. Thank you very much. Thank you everyone for joining this wonderful session, and I want to believe that we learned something. I want to believe that. And these things, I would really like it that even beyond this, one of the motivation behind programs that we carry out like this is that it can also help you to think wide in terms of research. You can think wide. There are different things you can research on. And when you get exposed to insightful webinars like this, you have some of these skills, it opens you up to creativity. And beyond all of this teaching, is actually that push. That's one of the things that is really pushing, that everybody that is able to partake of this can Mm. Okay. I think I lost my connection a bit. So please, can you hear me now?
Yes,
Can you
ma'am.
hear me now?
Yes, ma'am.
Yeah,
Yes, ma'am.
thank you. All right. Very shortly and quickly before we close, I still want to emphasize on the workshop, that I was talking about. Mm, let me see. Yeah. Okay. So let me just share it with us. And so for everyone that is interested, I don't know, can we still share the link currently on the call, or from our respective groups, more information will be dropped as touching it. So this is the workshop. This is the workshop. We are going to be revealing the hidden genetic gold mine in probiotics for next generation health industry and functional innovation. And the workshop highlights, we are going to be learning how to analyze probiotic genomes from the scratch. You understand the key functional genes that drive probiotic benefits. Yes, we'll understand some of these genes. We explore bioinformatics pipeline tools for functional prediction. And we are going to the hands-on workflow from assembly, annotation, and all of this workflow. They have different bioinformatics tools. We are going to explore them. And the date is actually going to be commencing from 15th to 18th of this December. Yes, it's going to commence from 15th to 18th, and the duration is 3 days. 3 days. The duration is going to be for 3 days between this space of 15 to 18. And of course, it's going to be online. Yes, the registration fee is valued at $100. Yes, but we are going to be reducing it, giving it at $25. So if you want to participate in this workshop, like now, you really want to participate, maybe you want to make part payments, you can reach out, and then we'll know what can work best for you. Of course, certification will be available. I think some people were asking about certificates for this webinar. We didn't prepare certificates for this webinar. Beyond the certificates, the main aim is the more insight, the more knowledge that you're going to be living. But certificate is not available for this webinar, but it's available for the workshop, of course, coupled with real skills that you can make use of. And the deadline payment is going to be 13th of December. So the curriculum overview, these are the things we are going to be dealing with on the day one. These are the things: foundations of probiogenomics, where you are introduced to probiogenomics, the importance, the databases that we're going to be making use of, the different tools we're going to be using. I talked about a tool that we use on that analyzing, detecting bacteriocin, and that tool is actually what we call Bagel. With the use of Bagel, we're able to know what type of bacteriocin is the particular bacteria probiotic producing. So that's what we're going to be exploring in day one. And then day two is now where we enter the functional gene discovery. So understanding the key functional categories of probiotics has to do with adhesion, immunomodulation, antimicrobial, stress resistance, gut interactions. And we are going to be seeing the genes that are responsible for this. We'll see the specialty genes. We are going to carry it out much more practically, and the safety evaluation, because for every probiotic there needs to be safety evaluation, checking that there are no virulent genes or AMR genes, safety genes, and all of that. And finally, on the day three, we'll be dealing with the interpretation, application, and innovation in terms of criteria for selecting probiotic strain. So after you've analyzed different strains, what are the criteria that will make you to choose a probiotic strain, the carrier relevance, even from this webinar, we've seen some of those relevance in terms of our career and practical relevance and all of that, and the application across diverse fields. So it's really going to be a packed workshop. So let me see how many people will be interested in that. If you are going to be interested, can I see some reactions if you would like to join this workshop, and you can also signify on whatever group that you are, those joining us from YouTube. I want to know, are you also on the groups? Any group that you are joining from, any WhatsApp group that you're joining from, or you are not in any group at all. Maybe you just came across this this webinar. Maybe you just came across it and you didn't have time to register before you joined. So please, so anyone, thank you. So you can just respond on the group, and we're going to share the details of how you can participate in the workshop. Yes, thank you. Another group has been dropped, so you can also help us to drop it on the YouTube channel, so that anyone that still wants to join, from the YouTube channel can join the group as well. So thank you, everyone. Thank you so much for joining. Thank you. We hope to see you in the in the workshop session, and beyond the workshop session, we hope to see you in further programs ahead, where we can make impact together, where we can share knowledge together, where we can innovate together. Because G-Natures, we have a lot. We have a lot that we are exploring, and we hope and we believe that indeed you can join us in this scientific journey, of discovery. We really hope to see you at the top. Thank you so much. Thank you, everyone. And have a wonderful rest of the day. So we are going to be ending the call now. So if you have other clarifications, we continue our chats on the WhatsApp platform. Thank you so much. Okay, goodbye, everyone. Goodbye.
Bye. Thank you.
Thank you so much. Okay.